I think we can get started. My name is David Fruman. I'm a professor in biosci and academic director of GPS STEM. Welcome you to the opening session of our workshop series on giving oral presentations. GPS STEM and before that GPS Biomed has invested a lot in scientific communication, but in reviewing our offerings, I kind of realized that they've been very focused on giving talks to lay audiences. So either the elevator pitch competition or the kind of performance workshops that Bree McWhorter gives about how to have stage presence and overcome nerves. And then there's Sandra Singh Low had a class for many years about the 5-minute TED Talk. But we really weren't providing the training on how to give your academic talk to experts or to scientifically trained individuals. And that's such an important part of your training, whether you stay in academia or go into just about any other field that you use your PhD. You really need to be confident and prepared when you give presentations. And that has personal meaning for me because, I always thought of myself as a pretty confident speaker and a pretty experienced speaker. And when I finished my postdoc, I had a lot of papers and I thought I was going to go out and get a job. And I interviewed, and my first 5 faculty talks didn't result in a job offer. And one of the faculty later on told me that my job talk really wasn't very well prepared. And I spent some time in his lab for a short second postdoc, and he helped prepare me to give a better job talk. And I interviewed here at UCI. I got the job, thankfully. All worked out in the end. And I've been here ever since. And I still work on my presentation skills. I know I always have more to improve. And I feel like I'd like to give back by having this workshop series. So I'm glad that you're all here. And I will like to open up, the first kind of 2/3 or 3/4 of this will be given by my colleague in molecular biology and biochemistry, Professor Melissa Ladone, who I always enjoy hearing her speak. And I was happy to welcome her to the class that I teach to first-year CMB students called PhD Fundamentals this year as a co-instructor. And she gave this lecture on oral presentations that I thought was very effective. So I invite her here to give this kind of general overview. And then I'll follow for the last 10 minutes or so about kind of some specific tips about the type of talk or how to prepare for that talk you give in 10 minutes at your science society, Society for Neuroscience, AI, whatever it might be, which pretty much everybody in the room, I hope will have a chance to do in your career, even if the pandemic has made it challenging to do that the last few years. So with that, thank you, Melissa. And
thank you, David. All right, thank you all for coming today. It's so nice to see some familiar faces. And to those of you who don't know me, as David said, my name is Melissa Ladone. I am a professor here in MD&B. I came to UCI in 2009. I can't believe it's been so long. As a new assistant professor, so just like David, I started my faculty career here at UCI. And I've been here ever since, and my lab works on innate immunity and host pathogen interactions and neuroinfection and neuroinflammation. So I really have enjoyed all of the colleagues who I have met here at UCI. And also I've really enjoyed training doctoral students and postdoctoral fellows and have a lot of trainees in my lab who we work on oral presentations with. And so today, as David mentioned, I'll just be giving kind of an overview to doing oral presentations, but I would really love to hear questions that you have that are maybe more field specific, because I think that one of your best resources is each other, right? So sharing with each other what you think is effective in your fields and ways to communicate with each other that really work. So I'm just going to start with a couple of questions. And I know for some of you who are, postdocs that this is I know the answer to this question, but so I don't know everybody here. How many of you have given a scientific presentation already? Okay, many, but not all. Okay. And did you receive specific training on that before you gave your presentation? Can you raise your hand if you did? Yes. Okay. Some of those who raise their hands are getting training. And what about feedback afterwards? Few. Okay. Okay. And what was most helpful from what you were told, either before or after your presentation? Or what resonated with you? What did you take with you to the next presentation that you had to give that you thought was useful? Anyone? Yes, go ahead.
So I think for our first year prelims, we had to give a scientific presentation on the paper that we read, and I did a mock one with my advisor, and something that there are 2 things he said. He said that I was being too specific about things that weren't important, and I could improve my visualizations. And I think those aspects really help me in my future. Like I make sure that my visualizations pretty much speak for themselves. And so that really helps.
Yeah, those are two really great pieces of advice. So one of the things that many of us struggle with is how much detail, right? We're in the minutia every day. And so it's easy to want to communicate all of that. But oftentimes that's not the most effective way to get your entire message across, right? And then the second thing is you're making all these slides. So you have a lot of influence on what they look like and how they're perceived by others. And so thinking very carefully about what you choose to put on your slides can be another very effective way to communicate. Okay, anything else? Any other thoughts or ideas? Yes, Rebecca.
We help the audience to understand what you're doing, give a clear message for each slide that you talk.
So a clear message for each slide. I think that's another really important component of putting a talk together is think about what the goal of each slide is, right? And that also helps with building the narrative of your story is if you can visualize each slide as part of a bigger story, then you can much more easily, I think, bring your audience through that story with you. There's another hand over here. Yes, go ahead.
One of my advisors told me that when I'm preparing for a presentation, I'm always going to be nervous, but when I'm really prepared, then the nervousness also comes with excitement to give that presentation.
Yeah, that's a really great way to help having the right attitude going into a presentation. For very few people, I think public speaking comes naturally. And so I think that one of the most effective ways that you can help yourself feel most comfortable is just by preparing for it and doing everything that you can to practice and be self-assured in what you're putting up on the slides. And then in addition, I think that this whole idea that if you think about your work that you're excited about and sharing it with someone, that's another way to feel less like you're being evaluated as you're giving a presentation and more like you're basically, I often try to think about it like, okay, I'm talking to a group of my friends. What do I want to tell them about my work, right? And how do I communicate that in a way that is going to resonate with them? Okay, what were some of the biggest challenges that you've been talking about preparing for a presentation with your advisor or your mentor? You raised one of them a moment ago, which is just nervousness, which is super common. That's good.
I think the biggest challenge is to seem natural for me because I'm an ESL speaker, and before my master thesis defense, my advisor said that, okay, you rehearse your oral presentation, but don't write all the script, don't write script and don't try to memorize it because while you are trying to memorize it, you will pause more than usual and it interrupt the flow of idea.
Mm-hmm. So trying to speak in what sounds natural, but sometimes if you have really short talk, you almost have to memorize it, but to not do it in a way that sounds like it's been rehearsed and memorized, right? That's a really good point. Yes, you had a point also.
Yeah, something I always struggle with is sort of, since we work in such a broad department with a bunch of different specialties, is how to simplify sort of the background and what the knowledge within sort of the rhetoric of my field without making it sound too simple, like I'm in too dumbed down.
Right.
It's finding a balance between those two features.
I think that you've hit on one of the biggest challenges with communicating science to a diverse audience, because you want to be able to speak to the aficionados and communicate that you actually know the nuance and the detail. But at the same time, you don't want to lose half your audience very early on in your presentation. So you need to also be able to speak to the kind of the bigger picture as well. And so I think that's part of where I think communicating with your PI about what are really those critical details that you want to make sure you include, and what can you actually leave out, or maybe leave for questions, right? And that's something that you'll work on as you go through science and give more and more presentations. Okay, so as we move on then, oops, sorry. So one of the things, and we don't have to spend as much time on this as I allotted, but think about a recent talk that you went to and maybe discuss with the people around you, what is it that you thought was effective about that talk? What did you like about it? And then also, what did you think they could have maybe done a little bit better? Or what did you take from it that you realized, you know, the next time I give a presentation, I want to try to avoid doing this, that, or the other. Just take a moment, speak with each other, and then we'll hear some of what you say. And feel free to rearrange yourselves if it's easier. And I'm going to walk around. So.
We were saying we like it when essentially people are able to sort of periodically throughout the talk, tie it back into why the talk is important. Sometimes you can sit there in 15 slides through, you're like, wait, why are we doing this again? And so it's creating checkpoints throughout to kind of bring the audience back in and let them pick back up. Oh, that's right, we're doing this. What's the next thing?
Right, so like reinforcing the goal as you move through the presentation. I think that's a great way of keeping your audience engaged and reminded when you've gone through 15 data slides of really what was the big picture question here. Sometimes people will use, oftentimes when we work on, biochemical pathways or signaling pathways, we'll have a model slide and we build it as we go. And so you have a series of data slides and then you go back to the model and now incorporate what you've now learned into that model, build more data slides and then incorporate, and then finish with the final model at the end, right? And so all the different ways that you can tie your story together in a cohesive narrative, I think is very helpful for engaging your audience. What other thoughts? Yes.
It's a fairly similar vein. We discussed how something we like is if people mention the specific reason that they did each individual experiment and like what question it was answering, because if you're like an expert in your field, then you know exactly, oh, this experiment's obviously the next one to do after we did this, but if you're kind of more adjacent, then it's not always obvious. And then you see all this data and you're like, I don't even know why you did that.
Yeah, so one of the things that you're hitting on is the transitions, right? Building transitions from one experiment to the next is a really effective way of bringing your audience with you. And it seems so simple, right? Just what is the conclusion of this experiment? Okay, and now what is the next question? And what did we do to test it, right? It doesn't take a lot of time, but it really helps people be able to follow from one set of data to the next, in particular with the narrative of why you're doing the experiments. That's a really great point. Other thoughts? Yes, Laura.
We were talking about how we like when a speaker is prepared and excited about their talk, and then something that maybe is not as ideal as when people have too much data and it can be a little bit overwhelming to understand the point, or when people don't explain their X and Y axes. And so someone outside of the field may be a bit confused about what they're looking at.
So being clear about what the data are that you're showing, and as simple as what are the X and Y axes. I think that's a great thing to note. I always tell my trainees when you're showing a graph, the first thing you need to explain is what the axes are, what the experimental groups are, and then describe the data. Because a lot of times people will point at a graph and say, you can see, and then they'll point to like a high bar and a low bar, and you don't even know what the graph is or what the different experimental groups are because they're not labeled in a way that's clear. And so then you automatically lost them. And so trying to be really clear about what you're showing in your graphs is really important. Okay, all right, why don't we keep going now? You've raised almost every point that I'm going to talk about, so I'll go through them a little bit more explicitly. But if you have other comments or questions, please feel free to interrupt. David has created these workshops for you, and so however they can be most helpful to you is going to be the best use of the time. So I don't know that I necessarily need to spend a lot of time on this because David gave such a great, compelling story about why it's important to take oral presentations seriously. But you're all being trained as some sort of scientist, and there are really very few career paths where you're not going to need to give probably fairly regular oral presentations. And even as part of your training, you're probably going to be doing this as well. And as you all know, those who have communicated their science effectively to you are those who you have remembered their work, you can build on it, you think about the kinds of conclusions that they're able to draw, and their findings resonate with you because you actually understood the presentation that they gave. And so if you can communicate your science effectively, you're just going to be a more competitive speaker. In the case of a job talk in particular, where there really might be only one person who's going to obtain the position, there are lots of opportunities for best talk awards or best dissertation presentation, things like that, that you can be competitive for if you've spent some time preparing your talk and really taking the time to do it well. And then various other types of opportunities as well. And as you share your data with others, you also gain from having an impact on your research through getting feedback about moving your project forward, potentially initiating collaborations. This is something that is one of the real benefits when you go to a conference and you present on your research. If someone perhaps at a neighboring institution is working on a similar project, and would like to collaborate with you because you're taking a complementary approach, like this could be a great way of expanding your network and really enhancing the impact of your work. Always getting new ideas and perhaps a new perspective on your work. So I'm going to go through 10 tips. Many of them are things that we've already talked a little bit about, but I think that this slide deck will probably be shared. You guys usually share that you get. So you can always go back to this later when you're actually putting together a presentation. The first is really finish within the allotted time, right? So in any opportunity that you have to speak, there will very likely be a clear time frame for it. So for instance, if you're giving a talk at a conference, you may be given a 30-minute time slot, but only 25 minutes of that is intended to be your talk, and then 5 minutes is question and answer. So you really need to finish at the 25 minutes, in part because if you're at a conference, likely you have a whole set of other speakers who are lined up behind you. And so conference organizers and session chairs don't take well to people who go way over their time and will very likely cut you off, right? And at certain types of conferences, like at Gordon conferences that I've been to, there really is like a machine sitting on the podium that has a green and a red light on it, and it starts flashing as you get closer to the end of your time, because they really want to preserve enough time to have question and answer and discussion. So you need to make sure that your talk is going to be finished within the time that's allotted. It's also just an indication of respect for all the other speakers who come after you, right? And so one way of planning for that is to estimate about a slide every minute, right? This is really going to depend on the nature of the talk you're giving and the content of your slides, but this is generally a good framework for structuring a talk. Some slides will be really quick. So your title slide, you're going to zoom through. You might have a slide that just has your hypothesis on it, and you'll state it and you'll move on, and it'll take 5 seconds. But your data slides are probably going to take more time to explain. And so on balance, about a slide per minute will generally work out. The second major tip is to really know who your audience is. This is super important when we think about those earlier comments that people raised about how much detail to provide, right? So you want to make sure that you are tailoring your background appropriately. When I was in the back of the room, one of the comments that was raised was, it's frustrating when you go into a talk and the speaker, in this case, it was in Department of Chemistry, and the speaker sort of dives right into data on the first couple of slides without really providing the necessary background for the entire audience to follow, right? Then you're going to spend an hour not necessarily understanding what the purpose of all of the work is, right? So try to tailor your background for the audience. And for instance, for let's say a 25-minute talk, you probably want to spend a good 5 to 10 minutes really building the scientific basis for your work. And so the background, the hypothesis, the rationale, the significance of the work, why are you doing this study? I know this seems like a lot of time to spend on background, but if you don't convince people that the question you're asking is important, it's going to be really hard for them to really appreciate all of the data that come afterwards. For a specialized audience, you can probably get rid of some of that background, right? If you're speaking to an audience of specialists who study cell death pathways in cancer cells, you probably don't need to explain what cell death is, right? And it's probably a waste of your time to do that. But you may be able to go into more details about all the different mechanisms of cell death and the specific pathways that are involved in cancer cells. For a general audience, this comes to the point that was raised earlier. It's probably okay to omit some of the technical details because the general audience isn't necessarily going to appreciate it. And this is a situation where the advice of your PI is going to be really helpful here because they have very likely given talks to both types of audiences and can tell you, you know, I really thought that this piece of information was critical for this audience to know, whereas some of these other details are maybe more superfluous. Okay. Tip number 3, why should they care? So it can be very easy to overlook this point because you have invested so much time and energy in your research. You certainly know why you care about it, but someone who's listening to you may not fully appreciate really the significance of the question, especially if they don't work directly in your field. They might not necessarily appreciate that there's a big knowledge gap that you're addressing. And so that should really be front and center. And of course, if you don't seem like you care about your research, it's really hard to convince other people to care about it, right? And so, for instance, if you're studying a disease, which many of us in the biomedical sciences do, talking a bit about the prevalence, the symptoms of the disease, the morbidity or mortality rates, who's affected, the epidemiology of the disease, that's going to help people connect with the kind of work you're doing because they'll be able to appreciate on a larger scale the importance of your work. In contrast, if you're working, let's say, on developing some new transformative technology, right? Really explaining the application of that technology, why it's needed, how it's an improvement over what people are currently doing. That's going to really help with people understanding why they should care. The second kind of related question is, why are you doing this project? So you may recognize that it's important to, for instance, develop a cure for a particular type of cancer. But within that, there are a huge number of different questions you could be asking. Why is it that the question you're asking is the relevant question, right? And so the rationale for your project should really build scientific basis for your question and hypothesis. And I think it's important to realize that not everyone's automatically going to appreciate the importance of your work unless you're explicit about it, really telling them why you're asking a good research question. And so earlier when I was kind of walking around, someone had said they really appreciated when people were giving talks and they took people kind of step by step through the logic and why they are taking the approach that they are. And I think that that's helpful because all of us can follow logic like that. And hopefully your audience will arrive at the same conclusion that you have, that this approach that you're taking is really going to be the best approach to answering this really important question. And then you have them all on board and engaged as you kind of go into your data. And so what is the overarching question or problem? What's the gap in knowledge? What do you hypothesize? And then how are you approaching this? It's really useful to have slides that kind of address these questions. They don't have to be titled as such, like this is the gap in knowledge or this is the question. But communicating this logic as you're moving through is going to help people really understand why it is that you're doing the research that you've decided to conduct. So a very standard framework is the one that's shown here. And this is not just for presentations. Many of you have probably, have any of you written fellowship applications? I know some of you have. Okay, so fellowship applications, if you write a manuscript of your scientific research, it follows a very similar structure. And so if you can build on a structure like this for your projects, it's going to help you in so many different facets of your communication. So again, reinforcing the background, the significance, the rationale of your work, what the hypothesis is, and the central question. So some work has a very clear hypothesis. It's incredibly hypothesis driven. Other work is going to be a little bit more exploratory, right? And so you may have work that is really asking a broad question, and that's okay as well. But I think stating what that question is in an explicit way will really be helpful for your audience as you begin to move into what your findings are. And then, of course, most of your presentation will be spent on the data, results, and figures, and we'll talk a little bit more about ways that you can go through those, and then conclusions and future directions. So my sixth tip, which gets to a point that was raised earlier, is to make sure that you simplify your data to an extent that it's understandable, but you also don't want to dumb it down to the point where, you know, an expert in the field is going to feel like, you know, it'd be really nice if we could see a little bit more data, right? So it's kind of finding that balance. But when you show figures, I think showing figures that are simple and clear and showing multiple slides of simple, clear figures is going to be much easier to understand than showing something like this. So this is just taken from a paper we published a few years ago from one of my graduate students. I really strongly advise you to never put a multi-panel figure on a slide like this when you're trying to communicate anything, really. This sometimes happens when people are doing journal clutter. That's when I often see it happen, is that it's not necessarily your work where you have a slide of each individual figure, but you've downloaded the paper, you've taken a screenshot, or you've downloaded the figure, and this is kind of how it shows up on the slide. And then the logic is just to walk through it. But it's so hard for people to appreciate really anything from a slide that looks like this, in part because it's just very hard to see what's happening. And there's so much going on, it's hard to know what to focus on, right? And what's important. And so, for instance, if you were to take even just this figure, so these are largely flow cytometry plots. So instead of something like this, if I were to explain that panel, I would first probably explain how I generated the data, right? So what we're doing here is we're looking at subsets of immune cells called monocytes from human blood. And these subsets can be identified on the basis of the proteins they express on their surface. So in this case, this monocyte is expressing a protein called CD14, which we can detect with an antibody that recognizes CD14 conjugated to a fluorophore. And if we take these cells and run them through a flow cytometer, we generate plots that look like this, where each dot is an individual cell. And on the x-axis, we're looking at CD14 conjugated to the FTSE fluorophore, and on the y-axis, a protein called an antibody that recognizes CD16 conjugated to this fluorophore. So each dot is showing different levels of expression of these two markers, and then we can gate on these populations with these numbers representing the frequencies of cells within each of those, right? And so even those of you who've never seen a flow plot before in your life now can actually understand what this is, hopefully, to some degree, right? Whereas if I had just shown you this and said, look, we have these different populations of monocytes, it would really not be anything. I mean, you'd have to be trusting me, right? Because I hadn't explained the data to you in a way that you could actually sort of understand it, right? So you can conclude that we have the highest frequency of inflammatory monocytes, which express CD14, high levels of CD14 and low levels of CD6, right? And so thinking about breaking your data down in a way that will be digestible to people, I think is really helpful, okay? And this isn't to say that every single person in the audience has to necessarily follow every single piece of data that you show from the beginning of your talk to the end. I think that that's a really high bar to meet because everyone's coming in with diverse levels of background in your talk, right? But if you can keep most of your audience engaged all the way through, and even if they can't understand every single data point, if they can understand what the graph is meant to be showing and what you're concluding from it, I think that that's a huge win, right? So another strategy that can be very helpful if you're showing a graph like this that's got a lot of different conditions and a lot of different dots, and what you're really focusing on is one condition, using things like red boxes or animating your graph so that you're only showing the conditions one at a time and then you build the graph when it's complicated is another very effective way of having people focus on what it is that you want them to pay attention to. Because if I show you the data like this, it's really hard to know where to look. There's a whole bunch of significant stars that are kind of spread out all over the place. But if what I'm telling you is in this condition, we're comparing infected to uninfected samples that are treated with a vehicle control, and each dot is a different blood donor. We're looking at the release of a cytokine called IL-1, and then you can at least be focusing where I want you to focus because I put this big red box around it. And many of us, when we get nervous, we start to sort of shake a little bit with the laser pointer. Sometimes you'll be in a presentation where the laser pointer is not very good. And so even though you're trying really hard to point out a specific group or condition, it's not effectively showing to the rest of the audience. So I think using things like boxes or arrows or other animations into your PowerPoint can be a really effective way of helping people hone in on what it is that you want them to focus on. So in addition to keeping figures simple and clear, trying to avoid confusing people with tangential information is generally helpful. You know every experimental minutia of each graph that you're showing. But your challenge as a presenter is to distill that graph down to its core essence and communicate that. Because really, your audience is not going to understand the hundreds of words, or not going to remember the hundreds of words that you spoke throughout your whole presentation. They're going to understand core ideas, right, as you move through. And so you really want them to take away the essential details for appreciating the data. And one of the things that I try to have as a goal when I'm showing data is to explain it to a significant enough extent that the audience can actually be interpreting the data as they're looking at it and not just having to take my word for it, right? So you want to, this is where telling people what's on the axes is telling people what the experimental groups are, which are the controls, which are the experimental conditions. That it empowers them and enables them to be able to interpret the data as they're watching your presentation and listening to it. And it also makes them actively involved in your presentation in a way that I think that they're going to be much more likely to remember what you said, right? Because they're actually processing it as you're presenting. So you want to include as much key information on the slide as you need to for them to do that. Yes.
Do you prefer if we ask questions throughout it or?
Yes, please.
Okay.
I'll just adjust this.
Do you have a personal limit for how many figures you like to throw on the slide at once? Because I know there are times where even if you break them up properly, it's still a lot going on. And unless the person is walking you through the plot that they're talking about, it's easy to get lost.
Yeah, so it's such a great question. So I go through this a lot with my trainees when we're preparing talks for a conference or an advancement or committee meeting. I personally don't like, well, so if you have something that looks like this is just one graph, but it's so many conditions and data points that I wouldn't put anything else. I would just have another slide. So if I were to really present this, I would present this in probably 6 slides. just because I think it's easier for people to appreciate one idea at a time. But sometimes it can be helpful if you have data for which it's useful to see the comparisons, to be able to, just like you said, build it, right? And so maybe 3 or 4 graphs that are not as complicated as this one on one slide would be okay. Sometimes people will do things and we'll sometimes do this where we make a little cartoon. We do this a lot when we have pathways where we're using a bunch of pharmacological inhibitors that hit a different protein in the pathway. And they've got like these acronyms that are meaningless to anyone else, where we'll have a cartoon of the pathway with like each of the inhibitors, and we'll have that as a big slide when we first introduce kind of the approach we're using. And then for each inhibitor that we show, we have the graph, but then on the side, we have a small version of the cartoon with the inhibitors, right? And so there are ways that you can use information that you present early on later by kind of making it small and putting it on the side, because you've already explained it. So it doesn't need to be quite as big as it was the first time you showed it. That's another way to kind of deal with complicated information. I think in that way that sort of, does that help at all?
Yeah, could I ask one quick follow-up?
Yes, of course.
Are you pro or against animations? I feel like that's kind of a point of contention that some people have.
Yeah, people have strong opinions about animation.
Don't have something bouncing.
Yeah.
You don't like.
Yeah. So my feeling, this is totally a style thing, right? So everyone has to kind of find their way. My feeling is you don't want stuff like your animation to be a distraction. So I prefer either it appears or it disappears. But I don't like it when things like spin in or like, like fade or things that are going to suddenly distract from like what it was that you were showing. So that's my personal, but you all have to find your own thing, your own style. Okay. Any other questions while we're stopped? Okay. Okay. I already went through all this, and that's what you want to try to avoid. So this is one of those do as I say, not as I do kind of talks. Okay. So another thing that can be helpful is to really think about engaging your audience by treating your audience as you would friends that you're explaining your work to, or colleagues who have a vested interest in the kind of work that you do and want to hear more about it, right? As opposed to a room of strangers that are all evaluating you. So this is something that I find very helpful to do, is just move away from the podium, because I think it's much more helpful to engage if you don't have a big podium between you and your audience. But again, that's going to really depend on each person and how comfortable they feel with moving around. My husband, when he lectures, he walks around the aisles. I mean, I think I'm not coordinated enough to do that in a lecture hall, so I don't really try to do that. So you have to kind of figure out what works best for you. I really encourage trying to look at the same screen as your audience. After coming out of a couple of years of Zoom presentation, there's a real tendency for people to want to talk at the screen that is on their laptop and use the mouse on their laptop, which means that they're looking at one screen and their audience is looking at a different screen. And I think that immediately disengages you from your audience. So I think if you can both be looking at the same screen, it's helpful.
What's your tip then for like hybrid settings?
Yeah.
Just being on
Well, so
Zoom.
this is hybrid right now. I think we're on hybrid, aren't we? Because I'm on Zoom,
You're recorded, but not,
but they can't see me.
they're not online.
Okay.
So say you have you'll have like your virtual audience and then you're here, so you want to engage, but you also want to engage with so you want your face kind of in the screen. It's always going to be in a weird angle.
Yeah, so I have to admit, I've not been in exactly that situation yet.
We're going to have a workshop on Zoom talks or
Oh,
hybrid
okay,
talks
cool.
later on.
Yeah,
But somebody who's
But
been
the
more thoughtful about that.
advertising. Yeah, okay.
I mean, I guess one thing that could maybe be done is you could have the computer and the podium set so that it's not far away, but is that kind of an equal, so that you could sort of treat the computer as another person, right? So that you're talking to your audience and to the person or the people on Zoom. I don't know. I guess that would be something that would be useful to hear tips from.
Because I find sometimes like you're in situations, you're in like a conference room. And you're like, you have your laptop on a table, so you're talking to a few people, but then you have a whole audience.
Yeah.
And then like it's gonna be awkward, I
Yeah.
think, regardless.
Yeah, I
So
think
do
you're
you
right.
try to, like, right, like lean down, I guess,
Yeah,
and just get by with what you can.
I've seen people do that occasionally when they have like committee meetings and maybe like 1 or 2 of the committee members can't make it in person, or for whatever reason isn't in person. And so they're on Zoom. And I do feel like what ends up happening a lot is you end up focusing on the people who are right in front of you, right? I don't know if maybe that's part of the drawback, but it would be useful to hear what the actual advice is. Any other Okay, and so these are things that you've probably already heard. Try to avoid standing and reading your slide. Try to avoid slides with huge amounts of text. These slides actually have kind of a lot of text, but really trying to avoid paragraphs of text, because what you don't want is your audience trying to read and listen to you at the same time, because then there's conflict, right? They don't really know where they should be focusing and paying attention. Speaking faster often happens if you want to communicate high energy, and I will sometimes get in my teaching evaluations, she seems to talk really fast when she gets excited about something. And so I have to kind of deliberately slow down because I know that that's not effective. But there are going to be natural tendencies that you have, and I think being aware of them is the first part of it, right? So that you can work on them. And finally, you're really the biggest stakeholder in your research. You care more about it than anyone else. And so I think recognizing that as you give a presentation is really helpful, because you're the one who has the most impact on being able to disseminate that information. Practice. This is something that we discussed already. The more you practice, the more comfortable you're going to feel. You can help to avoid with fumbling for your words, understand which slides will need a strategy. This often happens when I do practice talks with my lab before they go out to give a talk. They'll realize there's a handful of slides where when they transition to them, they suddenly don't really know the best way to get into the slide. And so practicing it gives you a chance to figure out what's going to be my strategy here? What is the first thing I need to tell people? And then how do I build that? Or should I break this slide up into 2 slides, or should I animate it in a way that gives me cues how to present it? It'll also help with giving you a sense of timing for your talk and the length and comfort with the animations, and then figuring out those transitions that help you to really tell your narrative. Oftentimes people will conclude with a summary or a model. I think this is super helpful. It helps you to remind the audience of the goal of the project, which hopefully you've been doing as you've been going through. So by the time they get to the end, it all is crystal clear to them. How did your work address the question? And then if you've been building a model throughout, finishing with that model can be really helpful. One of my former mentors used to have a conclusion slide, do acknowledgements, and then during question and answer, he put the model back up again. And so then people are looking at that as they're asking questions, and you can kind of use the model as he's answering questions. Future directions is a really common slide to include if you have a research in progress talk or a thesis committee meeting. It's not as common to see it at a conference. I think most people will conclude with a summary or conclusions. In part, you're limited in time at a conference. So this isn't generally where people will go with a conference. And then finally, you have the chance to acknowledge your PI, your lab mates, your collaborators, people at core facilities who helped you. It's really your chance to thank people who've contributed to your success. And so you really want to be gracious with those acknowledgements at the very end of your talk. The last thing that comes is always the question and answer. Most talks that you give will have a Q&A period. And oftentimes my students will say, you know, I can prepare every detail of my talk. I can be totally ready, but I get so nervous with question and answer because you have no idea what someone's going to ask you and how prepared you're going to be for it. And so I really recommend pausing for a moment, reflect on what the question is. Sometimes as it's being asked, you'll know exactly what your answer is going to be because it's something you've thought very carefully about. Sometimes you really haven't thought about it. And so you need to sort of process, and repeating back the question can be helpful both for allowing people who didn't hear it to hear what the question is, but also for helping you maybe process the important parts of the question. Eventually, it'll get stumped, and it's okay. And it's okay to just say, you know, that's a great question. It's not something I've thought about. We haven't considered that, but we really should take a look at it and move on. Don't feel like you have to apologize. Just acknowledge, right? This is a good question, and we'll have to think about it.
Yeah, don't try to answer the question with a different question just to say something.
Yeah, true too. Sometimes students will just start talking and talking and talking, and it doesn't actually answer the questions. So that's also nuts. So just in the last 2 minutes, because I want to make sure that David has enough time to give his tips on short presentation, a few cosmetic considerations. And this kind of comes down to style as well, right? So take what I say with a grain of salt, but this is just, you know, for what it's worth. I really recommend using professional fonts, right? So these are some that are considered sort of professional, mostly since not all fonts are available on all different types of computers. I would really avoid using rare fonts. And the reason for that is sometimes you'll go to a conference and they'll ask you to upload your slides onto a conference computer a day in advance or something, and then it's going to be projected up from the podium. And maybe you're a Mac person and it's going to be projected on a PC or vice versa. You may not have control over computer that's going to be projecting your slides. And so if you're using a rare font, it's going to convert to something else if that computer doesn't have it. So using a common font is helpful. I also recommend not using any smaller than about 18-point font for main text. This is 22-point font. And I wouldn't say it looks too small, but if you go much below 18, it starts to become difficult to read at the back of the room. And then finally, people have strong opinions about this font. When I inherited these slides from a predecessor, it actually said, never, ever use Comic Sans font. So I'm not gonna be quite as dogmatic as that, but just be mindful that people have opinions about it. Think about the background that you use. So really commonly people will use white backgrounds as the default because it's easy to see black text on a white background, and most graphs work well on a white background. Projectors can handle a white background. If you switch to a black background, it's okay. You just need to then make all of your text. So you're starting to lose axes. I'm sorry, you're starting to lose axes now. So you need to go in and like fix all of that, which just takes a lot more time and energy, right? And you might forget something. The one exception is I tend to think fluorescence images look better on a dark background. So we do a lot of microscopy. This is a brain that we imaged from a mouse. And I think that you can see the fluorescence a lot better when the slide is a black background. But again, this is a style thing. And so at the end, thank you for your time, and I'm happy to take any questions.
So, say you have just a couple of slides that have fluorescent images, so is it okay you can switch between them?
So that's what we end up doing. We keep everything else about the formatting the same. So the same font for the titles, same, either it's left justified or it's centered. We just swap the text from black to white when we do the fluorescence. But again, that's kind of a style thing. If it's distracting to you to go back and forth, then don't do it. Just, include a white background and then have the fluorescence.
Briefly had a point on your slide talking about the title of the slide, for it to be the conclusion.
Yeah.
And I really like that, and I've been given that advice too. I try to do that, but I really struggle with titles for the starting part of my presentation. Like I just say background or introduction, and I don't really know if that's okay or.
I think it's better to have a descriptive title. So your background has to be about something. Like, what are the bullet points that are on your slide, or what's the graphic? And I would make that the title. Or what's the goal of you showing that slide? Like, what's the information that you want to communicate? And you could use that as your title as well.
The information I'm trying to communicate is the background. So I wouldn't know.
But you have specific information in each of your bullets, right? About whatever process or pathway that you work on, or disease or technology. And I think that's what you could include as your title. And I'm also happy to talk with you if you ever want to just bounce ideas about.
Really
about it.
appreciate that.
Yeah. Did you?
Yeah, if you had just finished like making your slides, do you have like a streamlined process where you practice your talk to become ready for the?
Yeah, so when you say just finished, like right before you have to give the talk, or like you finished
Like you
them?
finished making like the slides, but you haven't practiced the talk yet?
Yeah, so how do you practice it?
Yeah. I
Is
guess
that what you're asking? Yeah, I would find a room or a screen, stand up and talk as if you're talking to an audience with looking at your screen. Like really practice it in as an authentic way as possible. Sometimes when my trainees, when they're giving their talks for the first time, they'll want to practice sitting down at like the conference table. And I'm like, no, you gotta stand up, because it feels different to stand and point than it does to kind of sit behind your computer. So I think as much as you can, being able to practice the way that you'll be giving the talk is helpful. And I think the other thing that's helpful is to be able to give your talk without having to use like presenter tools, because it may be that you're at a conference where your computer is over here and you're asked to stand over here and you don't have access to them, right? So make sure that you can give your talk without relying on the tools on your computer. Yeah.
Nice. Yeah, go ahead. Take your time. Yeah, if mine gets short, and that's fine. I actually probably
In
5
your
minutes.
opinion, how much text is too much text on a slide? So at least for like background where like, maybe you want to hit a few bullet points, I'll put some, but when it comes to like data slides, I like to sort of have the figures and then me talk through it so that the focus is kind of on me and not just the slide. Although like you mentioned, maybe that's wrong because people should be able to interpret the slide without you being there.
So I think your intuition is right on this. So I also agree that if you can just show the figure without a lot of text, I think that the standalone figure is really helpful. And maybe you just have a little bit of text, like oftentimes we have, because we do infection models, there's a certain day post-infection that we harvest our mice or our cells or whatever. And so I'll sometimes put that information just so that someone knows what the time point was, or what the strain of pathogen was that we used, or the strain of mouse that we used. Something like some of those like technical details that it's kind of helpful to know. But in general, I don't tend to recommend having lots of text on your data slides. The one time I sometimes will encourage students to put text is if they're having trouble explaining the workflow. And I'll say, just put a quick workflow with like 3 little text boxes with arrows. And it might help to remind you to explain the experiment and then show the data. And that can also be helpful for an audience, right? That if they're not familiar with that particular type of experiment, that they can kind of see, you don't want a detailed workflow, but like 3 little text boxes with some arrows can be helpful, with just like 5 or 6 words in each one.
I want to ask, how long did it take you until you were like comfortable speaking publicly? Like years or presentations? Yeah, I don't know.
I'm the wrong person to ask because I did a lot of music performing as a kid and just got over nerves at a young age. And so I get nervous when I'm talking to people I know. When I have to give a seminar in my own department, I'm super nervous. But when it's just a bunch of audience people who I don't know, then I'm different than most people.
So yes, that's different than most people. So I still get nervous. So when I first started teaching here, I was teaching in Biosci 3, 400 students, microbiology. I was like 4 pages ahead of the students in the textbook the first quarter I taught it. So super nervous, right? I had never even been in a classroom that big, I don't think. So I think the more you do it, the easier it gets. And for any given talk, definitely the more you've given it, it will be, by the time you get to like the last job talk or the thesis defense, it will seem so much more natural than.
How many runs do you like to do before?
So
Like, say you have a new talk, right? If you have to come in from a new angle, you're tying different projects together. I feel like I run into that now where like it's the tie together, right?
yes.
Or then every audience changes slightly.
Yep.
How many times, like, the information in and out, but those transitions and then your big picture and how you're going to apply that might change. How many run-throughs would you?
So I frequently am in this exact situation because now I'm presenting on work for my trainees. And so oftentimes it's new and there are frequently conferences I'll go to where I'm telling a story for the first time. I would say I make my slides. I probably do at least 4 run-throughs of it. The first run-through is almost twice over time because I haven't honed any transitions. I'm saying a bunch of extraneous things. I haven't figured out how to explain the graphs. And usually by about the second or third one, I'm feeling like, okay, I'm close to within time. I've gotten rid of the stuff that I don't really need to say. I maybe fixed some of the slides so that I can get through them faster. And then by the 4th time, I generally feel like, okay, I think I can go and do this.
So your approach is to just go at it for a full run through without like a outline. Do you pre-think it through?
Well,
Like silently?
you end up pre-thinking it when you make it.
Build it, yeah.
Yeah. But I don't know. That's me. That's my problem. I don't know what your.
Yeah, that's pretty good.
Yeah,
That's
I
pretty
would
good.
say, I don't always speak it out loud. I speak it in my head.
Yeah.
I feel awkward talking to myself for some reason with nobody around.
We need an
Yeah, you have to
audience.
go through it several times.
I'm also happy to stick around afterwards, but I want to make sure that David. So I don't go over my allotted
And
time.
even though I don't get nervous, I do try to get into a zone for 5 or 10 minutes before my talk where I try not to get people disturbing it, turn off my phone and all that, because you really need to be focused. And so before I start my talk, this is the kickoff event for a series of workshops. And, these 2, or this one is being led by faculty, but the others are likely to be led by students and postdocs with experience giving those types of talks, or recent faculty who have given the faculty talks that we're going to be scheduling. And there'll be smaller groups and, the dates haven't been set, but after we're done here, you'll get a request to answer survey about which of these workshops you want to participate in. The RIP talk, the ESAS seminar talk, the postdoc talk, the academic job talk, industry job talk, et cetera. And we really appreciate you answering the survey. So when you get when you want to go to a conference, and I know that in the pandemic, not everybody's been able to go to their society conference, but hopefully that's changed and you're all going to be able to try to go give a talk. You might get stuck doing a poster along with the 10,000 other people in this huge room. But if you're lucky, you're going to get this nice conference email and you're going to be really excited that you got chosen for a platform presentation. But then 5 minutes later, you're going to start to worry about it. And my first tip, and I independently just came up with a series of slides that have tips on them, is to relax. And that's because this is really a great opportunity and it's very unlikely that anything serious is going to go wrong. More likely that this is going to be a terrific experience for you. It's actually a lot easier to prepare a 10-minute talk because you just throw out all the extraneous stuff. There's a very simple format that I will give you here. And also, the people in the audience are self-selected to be interested in what you're talking about. I don't know how many of you have gone to conferences, but typically I or anybody else selects which of the short mini symposia they're going to go to based on the overall content of the interest or because they did a search in the conference app for a keyword. And they came to your talk out of the 4 or 5 in that session because they want to hear what you have to say or they're interested in that general field. That also means they tend to be experts. And that means you don't have to worry about as much background or defining terms or even a lot of the kinds of approaches that you're using. They want to hear what you've done and what the conclusions are. And so it's a little bit easier. It's the simplest talk, I think, to give in many ways because it is not a general audience. And if you do well, you're going to be super happy. Your PI is going to be happy. The audience is going to be happy. They will have learned something. And you could even have a kind of a memorably good experience, like the first time I gave a talk at the AI when I was a third-year graduate student. I was working on immunosuppressive drugs, trying to understand their mechanism. And we had this hypothesis that calcineurin phosphatase was the target of cyclosporin A. And we converted a biochemical assay into a cell lysate-based assay where we could measure phosphatase activity. I went through the assay and how we could apply it to cells treated with cyclosporin. And then I got the questions at the end, and somebody in the audience said, so can you apply this Frumin assay to this other system? And I had like 4 people from my lab in the audience, and they thought that was the funniest and greatest thing. And they just, for the next 4 years, it was the Frumin assay, and I felt a little proud about it. I never thought of naming it after myself, but somebody did for me. That was a little, still remember, and I'm telling all of you today. So good things can happen in these short talks, but maybe you will go overtime, or maybe your slides, you'll be nervous, or you will answer a question, or you feel it didn't go quite as well as you wanted. No one's going to remember because they're going to go to 20 talks at this conference. And even the ones that they're disappointing to them, they're just going to be washed out of their memory. Maybe they'll start looking at their phone and they'll just forget about it. You still put this on your CV. So there's really no downside to getting selected for a short talk, and you don't need to stress about it. Just put in the time and you're going to do well and you're going to be glad that you did. And the same is true for even a kind of a smaller conference, like a Gordon conference. People are there, they understand the field, they want to learn something, but we've all gone to those conferences and 2 minutes later, I can't remember which were the disappointing talks, unless they're like a senior person who just completely wasted my time. And then you're like, wow, I didn't expect that from Professor X. Okay, limit the background. You don't have much time. So you really don't have, just do like 1 or 2 intro slides to focus on what's important about this talk, the problem, how the new approach you might have, and then use your time to present data slides in depth using the tips and tricks that we heard from Professor Ladone. And usually you can finish with a single summary slide, no future directions, and then your acknowledgements. I will say that the title that you put into these these platform presentations is important in a different way than if you're going to go out and give a seminar later in your career where you're just trying to give a catchy, snappy title that'll bring people, out of their labs to your talk. Here you want to include keywords that'll help people find you in all the different possibilities they can go to. So put keywords into your title that will draw them to come to your talk based on a search. I don't think I have to go into this too much, the significance. Even though this is a specific audience, you really need to focus on why this work is important. And you know, a lot of these tips and tricks I'm giving you today were adapted from a list that my colleague, Amy Ettinger, wrote up for our training program, Cancer Biology. This is a picture of Amy here, and it really kind of summarizes a lot of what we heard before. You know, why should they care? Why do you care? Why is this important? I will say another tip that I didn't put from Amy is that audience doesn't usually include cats, so don't wave the pointer. So we heard about the laser pointer issue. And then data quality. I shared this with Joanne earlier on. I'm saying, you know, you only want to show your best data. And I think that came out wrong. I wasn't trying to say you want to cherry pick experiments that worked and forget about the experiments that don't. You only want to give this talk if you're confident in the project and your results. When I talk about data quality, what I mean is that your images and the way you've marked them up are pleasing to the eye. And we saw a little bit of that from Professor Ladone. Here's some examples. Again, I took this image on the left from one of Amy Ettinger's paper, and you know, her lab are experts in microscopy. They have a fluorescence microscope in their lab. They're looking at this punctate staining. You can see the nice dappy nuclei in these cells. And then this is from somebody in my lab who was a novice trying to get you know, fluorescence microscopy to work, and I'm not an expert either. And he came to me, and you know, the different panels have different numbers of cells, different sharpness. Sometimes you can see the nuclei in blue, sometimes they seem to be purple. You get the point. You know, this is not an image, even if the data were spectacular, that you want to show to anybody in your field. So make sure that you have them nicely presented. Here's another cute thing that I got from Amy Ettinger, which is, this is what this abbreviation is. And so, are there any cancer biologists in the audience? So, okay. So even in the specialized audience of people who know about whatever cancer you're working on, I bet you that you can tell me what TNBC stands for, or AML. I'm not going to put you on the spot. But for those who are not cancer biologists, these are universally used for triple negative breast cancer and acute myeloid leukemia. If your lab is a world expert in JMML, you might think all the people who come to your talk will know that, but most cancer biologists actually don't know what that is. It's juvenile myelomonocytic leukemia. It's very, very rare. It might be passion of your lab, but this is an example of even a specialized audience, be careful about abbreviations. And lastly, this is kind of reinforcing what we heard before. But when you end with a model slide, the temptation is you know, I don't feel like using BioRender or, God forbid, PowerPoint. I'm just going to throw all my thoughts onto the wall. And so, you know, people can look through this, and I think they get a pretty good idea of what you're trying to say, but I think it's a lot easier to get the message across, and they'll remember it better with an image. And that's, you know, that's kind of a humor here, but here's an example of a talk I might give. And these are the conclusions of my talk of this kind of hypothesis that we've been trying to test. But it's much better to just present it in a model figure like this. And I can go through and say all these things that you see on the right, and I can even animate them in if I want to, as I go through the different points of this figure, as I try to make these points. But a lot of people are visual learners, and they'll actually be happy that you presented them with a model that they can look at instead of having to read as you repeat what you've said there. So these are just some general tips for the short talk. I tried to keep it brief within my 10 minutes. I think I succeeded. We can go over time. And originally I wanted to talk about answering questions, but we heard about how to answer questions. So I will just be the one to answer the questions. And thank you for your time. Yes.
Not necessarily for a conference, but like for a RIP talk, would the future direction slide go after or before your conclusion slide?
I would probably put it after the conclusion slide. Yes.
I know that we want to limit the amount of text that we put on the slide, but I also have other people's suggestion to give a statement on your title slide regarding the result that you find. Do you think that is helpful for the audience to understand the message?
So you say, helpful to make the title, the title of the slide a description of the result? Is that what you said?
Yes.
Yeah, very much. I think, but it's okay to have another like animated conclusion at the bottom if you want to rephrase it or just can even use it to add enthusiasm. Like this means that we cured mice from toxoplasma, you know, like not overuse that kind of thing. But we heard that comment before from somebody, that they were advised to make sure that your data slides, the title describes the result. And that can help, especially in longer talks where people tend to lose focus and it's easier for them to just read the title and kind of follow along. And it's interesting that this came up because this was one of the main weaknesses in my job talks, it turns out, is that, you know, a simple change of a descript, kind of a general title, analysis of phosphorylation of this protein in these cells, turned into this protein is decreasing phosphorylation by this. I could have gotten a job a year earlier, you know, retired a year earlier. But that one little, that actually was a big problem with my data talk. I will say that this issue about, you know, you want to have enough background. You don't want to have too much either because scientists want to see data. And so I've actually criticized in my first department talk at a retreat here at UCI, where I thought it's a general audience, I want to make sure they're all on board. And I only had 15 minutes and I spent 7 minutes on background. And somebody who had been at the bar right before kind of like spoke up and said, Fruman, get to the point, you know? So there's a balance there, yeah.
Detailed question about the data interpretation. If a figure or a panel belong to this figure is done with a specific method or technique, is it necessary to spend more time to explain this technique or make an additional slide to explain this?
I think it depends how much time you have. I think that if it's not a technique that is generally known by that audience, it's good. It really works well to have an intermediate slide where before I tell you the data from this experiment, I just want to make sure that I want to introduce the method and some of the key features of this method or key innovations. But if it's flow cytometry and the audience are immunologists, you don't need to explain it. You can just say the gating strategy, this. So it depends on the audience and how novel the technique is, I would say. Is that what you would, how you would answer? Melissa?
Yeah, I completely agree.
Regardless of.
This
the
is the
audience.
tailored for your audience.
If like we mentioned this before, like if the audience is like mixed, where you have people who don't know and people who do know, for example, like a thesis defense or something. So how do you do that balance? I think I wasn't really, like I didn't get that. Like I don't know how to do that.
I think that you should go to the thesis defense, the workshop. I would say for me, the thesis defenses that work the best are those that spend about 5 minutes giving the overview for people who are their mothers and fathers and cousins and brothers and significant others who know nothing about science. And then you just forget they're there and you talk to your department. But you talk about one of the 8 projects that you've been working on, not all 8. And then at the end, you tie it back to something that everybody can understand. And you finish in well under an hour because you need time to show embarrassing photos of your PI and, your dog and everything else, So, the thesis defense, a lot of people attempted after 5 years to show everything they've ever done. And it's really just pick the best vignette from all that time and show excitement about what you've achieved.
I guess having a conclusion as a title also helps balance out, like, even if they don't understand what I'm exactly saying, at least they know what the point of the slide is. I guess that's another way. Okay.
Everyone reads the title, right? So
Yeah.
even if all they do is read every slide title, they'll at least come away with all of the.
I think there was another question over here that I raised.
I think I had one, but so you had mentioned sort of tailoring the talk to your audience, which absolutely makes sense. Is this kind of format like it seems like a lot of the discussion has been about conferences and giving academic talks, but while I'm sure many elements are the same, are there any differences that we should note for, say, giving a talk to someone in industry?
You have
Like,
to go to the industry training
yeah.
session.
Oh, there's a workshop on that?
Yeah. some kind of cool border of the slide. And some drug, shape that's touching a cell or something.
I mean, the answer might be no. It might just be, it follows the exact same sort of flow that an academic talk would be. I'm not too sure.
I am not going to be the one giving that talk, but I'm going to try to get somebody who has that experience. I would say that industry people, they also care a lot about the big picture, but they want to see how you think as a scientist. Just as anybody hiring you for a faculty position or a postdoc would as well. That you had some problems and how you addressed them and what the cool things are that you achieved at the end of that struggle. And what you worked on isn't necessarily that important for what you're going to be working on in the company. That's a misconception a lot of people have, is that I am a neuroscientist working on ion channels, and therefore I can only get a job in a company that's studying ion channel blockers. They're looking for people who have good analytical skills, project management skills, who've worked, with people that have mentored people, that have done collaborations. And so in the industry talk a lot about collaborations, talk a lot about the resources you use to learn about. You went to a workshop to learn about bioinformatics or microscopy. So that shows that you know how to navigate science and work with people and fill your gaps in knowledge.